Good morning, everyone. Today we'll be diving into a core concept in immunology, the interaction between antigens and antibodies. This is one of the most fundamental processes underpinning how our immune system detects, targets, and eliminates foreign pathogens. We'll cover the theory, mechanisms, types of reactions, and important clinical applications. By the end of this lecture, I want you to be able to define what an antigen and an antibody are, explain how they interact, differentiate between the types of antigen-antibody reactions, and understand how we use these reactions in diagnostic settings. These principles are critical both in understanding immunity and in many medical technologies. Let's start with a broad overview. Antigen-antibody interactions are not just textbook immunology. They're at the heart of diagnostics like COVID-19 rapid tests and autoimmune screenings. When an antigen enters the body, the immune system's response is to generate antibodies, specific proteins that can bind to that antigen and help neutralize it. Let's begin with the antigen. An antigen is any molecule that can be recognized by the immune system and elicit an immune response. Typically, these are components of pathogens, like bacteria, viruses, fungi, or even parasites. However, antigens can also include non-infectious substances such as pollen, food proteins, or even self-molecules in autoimmune disease. Most commonly, antigens are proteins or polysaccharides due to their structural complexity and diversity. The part of the antigen that is actually recognized and bound by the antibody is called an epitope or antigenic determinant. Each antigen may have multiple epitopes, meaning it can bind to multiple antibodies, possibly simultaneously. This multi-epitope nature allows for a stronger immune response. For example, the surface of a virus like influenza is rich with hemagglutinin and neuraminidase proteins, each with multiple epitopes that our antibodies can recognize. Antigens can be classified as exogenous, originating from outside the body, like microbial toxins, endogenous, produced within cells, like viral proteins in an infected cell, or autoantigens, normal self-proteins mistakenly targeted by the immune system. Another term you'll hear is hapten, a small molecule that is not immunogenic on its own but can become antigenic when attached to a larger carrier molecule, often a protein. This concept is critical in drug allergies, where small drug molecules act as haptens. The ability of an antigen to induce an immune response is called immunogenicity, and this depends on factors such as foreignness, molecular size, complexity, and the host's genetic background. In summary, antigens are the targets recognized by the immune system, and understanding their structure and diversity is essential to appreciating how antibodies function. Now that we know what an antigen is, let's look at the molecule that binds it, the antibody or immunoglobulin. Antibodies are glycoproteins produced by plasma cells, which are differentiated B lymphocytes. Their primary function is to recognize and bind to specific antigens with high affinity and specificity. Structurally, an antibody is shaped like a Y. The two arms of the Y are called the Fab fragment antigen binding regions. These parts bind to the epitope on the antigen. The base of the Y is the Fc fragment crystallizable region, which mediates the antibody's effector functions, such as binding to Fc receptors on immune cells or activating the complement system. Antibodies are made up of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains, held together by disulfide bonds. Each chain has a variable region at the tip, which is responsible for antigen binding. The unique combination of amino acids in these variable regions gives the antibody its specificity. There are 5 major classes or isotypes of antibodies: IgG, IgA, IgM, IgA, and IgD. IgG is the most abundant in serum and provides long-term immunity. IgA is found in mucosal areas and secretions like saliva and tears. IgM is the first antibody produced during an immune response and is excellent at forming immune complexes. IgA plays a role in allergic reactions and defense against parasites. IgD is mostly found on the surface of immature B cells. The specificity and adaptability of antibodies are what make them ideal for use in both diagnostics and therapeutics. The antigen-antibody reaction is central to humoral immunity and forms the basis of many diagnostic tests. This reaction involves the binding of the antibody to a specific epitope on the antigen. It's important to note that this binding is non-covalent, meaning it's reversible and based on multiple weak forces, such as hydrogen bonds, electrostatic interactions, hydrophobic forces, and van der Waals interactions. The strength of this binding is influenced by the fit between the antigen's epitope and the antibody's variable region. Think of it as a lock and key mechanism, but not a permanent one. Because of the reversible nature of the binding, the reaction can be disrupted by changes in pH, temperature, or salt concentration. Once the antigen-antibody complex forms, it can trigger several downstream immune processes: neutralization of toxins or viruses, opsonization for enhanced phagocytosis, activation of the complement cascade, or agglutination of pathogens. This interaction can be detected and visualized in the lab using various techniques. The simplicity and specificity of the antigen-antibody reaction are what allow us to develop tests like ELISA, lateral flow assays, like pregnancy or COVID-19 tests, and even Western blots. It's also worth noting that the same principles apply in therapeutic antibody design. For example, monoclonal antibodies used in cancer immunotherapy or autoimmune diseases rely on this specific and strong antigen binding. Several key factors influence how well an antibody binds to an antigen. First is affinity. This is the strength of a single interaction between one antigenic epitope and one antibody binding site. The higher the affinity, the stronger and more stable the complex. Then we have avidity, which is the overall strength of binding when multiple antigen-antibody interactions are considered. For example, IgM has 10 potential binding sites. Its individual binding affinities may be lower than IgG, but its avidity is much higher due to multivalency. Specificity refers to the ability of an antibody to distinguish between similar epitopes. A highly specific antibody will bind only to its target antigen and not to structurally similar molecules. This is crucial in diagnostics, where false positives due to low specificity can be misleading. Conversely, cross-reactivity occurs when an antibody binds to an antigen that shares a similar epitope with its intended target. This can sometimes be beneficial, as in vaccines where one strain can offer cross-protection, but it can also lead to pathological conditions. A classic example is rheumatic fever, where antibodies against Streptococcus pyogenes cross-react with heart tissue. Other factors that affect binding include pH, temperature, ionic strength, and the time of incubation. All of these must be carefully controlled in diagnostic assays to ensure accuracy. Understanding these factors helps not only in appreciating the biology of immunity, but also in designing more sensitive and specific laboratory tests. One interesting phenomenon in antigen-antibody reactions is the concept of the zone of equivalence. This refers to the optimal ratio of antigen to antibody, where large immune complexes form that are visible, for example, as a precipitate in a gel. There are 3 zones to understand here: the pro zone, the zone of equivalence, and the post zone. In the pro zone, there is too much antibody relative to antigen, so no lattice formation occurs because the antibodies are saturating the antigen epitopes. In the post zone, there is excess antigen, and again no lattice forms because each antibody binds to only a few antigens without cross-linking. Only in the zone of equivalence, where antigen and antibody concentrations are balanced, do we get optimal cross-linking and visible precipitation. This principle is important in immunodiffusion tests like the Auchterlonie assay, and even in some rapid tests, where incorrect ratios can lead to false negatives. Clinically, prozone effects can occur in tests for syphilis or brucellosis if patient antibody levels are too high. It's a classic example of how more is not always better in immunology. There are several types of antigen-antibody reactions, and each forms the basis for a distinct laboratory method. Let's briefly overview the most important types. Precipitation reactions. These involve soluble antigens and antibodies forming in soluble complexes. Think of double immunodiffusion. Agglutination reactions. These occur when particulate, insoluble antigens, such as cells or beads, are clumped together by antibodies. Blood typing uses this principle. Complement fixation test, or CFT. an older method, but still important historically. It tests if an antigen-antibody complex can fix complement. Neutralization. This refers to antibodies blocking the biological activity of a toxin or virus. Vaccines aim to stimulate such antibodies. ELISA, enzyme-linked immunosorbent assay, widely used to detect either antibodies or antigens in a sample. We'll cover this in more depth. Immunofluorescence, fluorescent-labeled antibodies used for detection in tissues or cells under a microscope. Western blotting combines gel electrophoresis with antibody detection to identify proteins, used for confirmatory HIV testing. Each reaction type has its ideal use case, advantages, and limitations. As you move into clinical practice or research, knowing when and how to use these tools becomes essential. Precipitation reactions occur when a soluble antigen and its specific antibody interact in optimal proportions to form an insoluble complex. These reactions are foundational in immunology and were among the earliest studied laboratory methods for detecting antigen-antibody interactions. The key point here is that both antigen and antibody must be soluble, and the reaction is visible as a precipitate, which can appear as a line or cloudiness depending on the method. The mechanism is based on lattice formation, where the multivalent antibody binds to multiple epitopes on multiple antigen molecules, creating a large network. When the antigen and antibody concentrations are just right, in the zone of equivalence, this lattice becomes large and insoluble enough to fall out of solution. There are several types of precipitation tests. Radial immunodiffusion. Mancini method antibody is mixed into a gel, and antigen diffuses radially from a central well. The diameter of the precipitin ring correlates with antigen concentration. Double immunodiffusion. Auchterlonie both antigen and antibody diffuse through agar gel. Where they meet in optimal proportions, a precipitin line forms. This is especially useful for comparing antigenic relationships. Immunoelectrophoresis. A combination of electrophoresis and immunodiffusion to separate and identify proteins. These techniques are largely qualitative but are extremely valuable in identifying fungal infections, certain autoimmune disorders, and even immunoglobulin deficiencies. The key learning point is that precipitation reactions visualize antibody-antigen complexes only when conditions are ideal, reinforcing the importance of concentration balance and specificity. Agglutination reactions differ from precipitation reactions in that the antigens involved are particulate, not soluble, typically whole cells like red blood cells or bacteria. When antibodies bind to these cells, they cross-link and form visible clumps. This reaction is quicker and more sensitive than precipitation and is widely used in diagnostic labs. There are two major forms: direct agglutination and indirect or passive agglutination. In direct agglutination, the antigen is naturally present on the particle. A classic example is blood typing, when anti-A or anti-B antibodies cause agglutination of red cells with A or B antigens, respectively. This is also used in bacterial identification, like the Weidel test for salmonella infections. In indirect agglutination, soluble antigens are artificially attached to carrier particles, latex beads, or red blood cells. This extends agglutination to reactions that wouldn't normally occur. For instance, latex agglutination tests for rheumatoid factor or CRP use this method. Another variant is reverse passive agglutination, where antibodies are attached to particles and used to detect antigens, such as in meningitis diagnosis. Agglutination reactions are fast, cost-effective, and easy to perform without special instruments. They form the backbone of many point-of-care diagnostics. However, they require careful interpretation. Pro-zone and post-zone effects can cause false negatives, and nonspecific agglutination can lead to false positives. Understanding the principles and types of agglutination is essential for interpreting results correctly, especially in blood banking, infectious disease testing, and autoimmune workups. The complement fixation test, or CFT, is a classic immunological assay that relies on the principle that antigen-antibody complexes can activate and fix complement proteins, a group of plasma proteins that assist in immune defense. Though it's now mostly replaced by ELISA and molecular tests, CFT has historical significance and is still useful in some viral and parasitic disease testing. The test has two stages. In stage 1, patient serum is mixed with a known antigen and complement proteins. If the patient has antibodies against the antigen, immune complexes will form and fix the complement. In stage 2, sensitized red blood cells, sheep RBCs coated with anti-sheep antibodies, are added. If complement was fixed in the first step, there's none left to lyse the RBCs, and the cells remain intact. This is considered a positive test. If no antibodies were present originally, complement is still available and lyses the RBCs, leading to hemolysis, which is a negative test. CFT is mainly qualitative and was widely used to diagnose infections like syphilis, Coxiella burnetii, Q fever, and Mycoplasma pneumoniae. It's also used in veterinary medicine. The test is highly specific but technically demanding, requiring fresh complement, careful timing, and multiple controls to avoid nonspecific lysis. One limitation is that it cannot detect antibodies of certain isotypes like IgA or IgM very effectively. Understanding CFT helps appreciate how immune complexes trigger downstream responses and how we can harness these reactions for diagnostic purposes, even if newer tests are more commonly used today. Neutralization reactions are some of the most biologically relevant antibody functions. In these reactions, antibodies bind to pathogens or toxins in a way that blocks their biological activity. This is a key mechanism of protection in many viral infections and forms the basis for many vaccines. There are two main types of neutralization. Virus neutralization, where antibodies prevent viruses from attaching to or entering host cells. This is the goal of many antiviral vaccines, like those for polio, influenza, or COVID-19. Neutralizing antibodies typically target viral surface proteins, e.g., spike protein in SARS-CoV-2. Toxin neutralization, where antibodies bind to bacterial toxins like diphtheria toxin or tetanus toxin, preventing them from damaging host tissues. In laboratory settings, neutralization tests can assess whether a serum sample contains antibodies that can block a pathogen's activity. A common format is the plaque reduction neutralization test, or PRNT, where the reduction in viral plaques on a cell monolayer indicates the presence and potency of neutralizing antibodies. These reactions are functionally significant because they don't just label pathogens, they directly stop their function. Monoclonal antibody therapies like palivizumab for RSV and convalescent plasma therapy both depend on strong neutralizing antibodies. Neutralization is also important in passive immunity, for example, maternal IgG antibodies that neutralize pathogens in the newborn. In clinical immunology, measuring neutralizing antibody titers can help determine vaccine efficacy or past infection. Overall, neutralization reactions exemplify the immune system's precision, not just recognizing threats, but disabling them. The enzyme-linked immunosorbent assay, or ELISA, is one of the most versatile and widely used immunological tests in both research and clinical labs. Its main strength lies in combining specificity with sensitivity in a format that is easy to automate and scale. ELISA can be used to detect either antigens or antibodies in a sample. The basic principle involves immobilizing either an antigen or antibody onto a solid surface, usually a 96-well plate, adding the test sample, then detecting binding using a secondary antibody conjugated to an enzyme like horseradish peroxidase, or HRP. A substrate is then added, which changes color if the enzyme is present, providing a quantitative or qualitative readout. There are 4 common ELISA formats. Direct ELISA detects antigens using a labeled primary antibody. Indirect ELISA detects antibodies. A secondary labeled antibody is used for detection. Sandwich ELISA uses a capture antibody to bind antigen, then detects with a second antibody, ideal for antigen quantification. Competitive ELISA measures antigen or antibody by competition with a labeled version. ELISAs are used to detect HIV antibodies, hepatitis B surface antigens, allergens, hormones like HCG in pregnancy tests, and cytokines. They're also used to evaluate vaccine responses. Results can be read using a spectrophotometer, and high-throughput automation is common in diagnostic labs. ELISA's main advantages are its adaptability, quantitative potential, and high specificity. However, it requires multiple steps, and background noise can affect sensitivity. Mastering ELISA techniques is essential for any immunologist, clinical scientist, or healthcare worker involved in diagnostics. Immunofluorescence is a powerful technique used to detect specific antigens in tissues or cells using antibodies labeled with fluorescent dyes. It's especially valuable in pathology and cell biology, allowing not just detection, but localization of target molecules under a fluorescence microscope. There are two types. Direct immunofluorescence, or DIF, a fluorescent-labeled antibody binds directly to the antigen. Indirect immunofluorescence, or IIF, an unlabeled primary antibody binds the antigen, followed by a fluorescent-labeled secondary antibody. IIF is more sensitive due to amplification. In clinical practice, direct immunofluorescence is used in skin biopsies for autoimmune blistering diseases like pemphigus vulgaris, where antibodies target skin adhesion molecules. A positive direct immunofluorescence shows intercellular or linear basement membrane fluorescence patterns. Indirect immunofluorescence is used in ANA testing for systemic lupus erythematosus, where HEP2 cells are used as substrate and patterns such as homogeneous, speckled, or nuclear provide diagnostic clues. Immunofluorescence can also be used in microbiology, for example, detecting Plasmodium species in malaria or chlamydia in cervical smears. The technique's strength is its ability to combine antigen detection with spatial information, showing where in the cell or tissue the antigen resides. However, it requires skilled interpretation, a good microscope, and careful controls to avoid nonspecific fluorescence. It remains a cornerstone in immunopathology and autoimmune diagnostics. Western blotting is a widely used technique that allows us to detect specific proteins in a complex mixture using antigen-antibody interactions. It's especially important in confirmatory diagnostic testing, such as for HIV, Lyme disease, and certain autoimmune diseases. The process involves three major steps. Electrophoresis. Proteins are separated by size using SDS phage, a polyacrylamide gel. Blotting. These separated proteins are transferred onto a membrane, nitrocellulose or PVDF. Detection. The membrane is incubated with a specific antibody against the target protein. A secondary enzyme-linked antibody is then added, and substrate produces a signal, usually colorimetric or chemiluminescent. The advantage of Western blot is its specificity. It can confirm the identity of a protein based on both its size and antibody recognition. In HIV diagnostics, for example, it confirms the presence of antibodies against specific viral proteins like GP120 or P24. It also plays a major role in research, identifying protein expression, post-translational modifications, or effects of gene knockouts. While powerful, Western blotting is labor-intensive and semi-quantitative. It requires careful controls, good electrophoretic separation, and optimized antibodies. Interpretation can be subjective, especially in borderline or weak bands. Mastery of Western blotting gives students a solid foundation in molecular immunology and diagnostic technique development. Now let's delve into two key concepts that often cause confusion but are essential to understanding antibody binding: affinity and avidity. While these terms are closely related, they refer to distinct aspects of how antibodies interact with antigens. Affinity refers to the strength of the interaction between a single antigenic epitope and a single antigen binding site of an antibody. It's a measure of how tightly an antibody binds to its specific epitope, often described using equilibrium constants. High-affinity antibodies form strong interactions even at low antigen concentrations, making them more effective in neutralization or detection. In contrast, avidity is a measure of the overall strength of binding between a multivalent antigen and a multivalent antibody. Think of avidity as the cumulative binding strength when multiple binding sites are involved. Even if each individual interaction affinity is weak, the overall binding can be strong due to multiple points of attachment, much like how Velcro works. Let's take an example. IgG antibodies are typically monomeric and have high affinity, but lower avidity compared to IgM. IgM, being a pentamer, has 10 antigen binding sites. Each site may have moderate affinity, but the total avidity is very high. That's why IgM is particularly effective in early immune responses when antigen levels are high. Avidity also plays a critical role in diagnostic tests. High avidity antibodies are less likely to dissociate, ensuring more stable results in assays like ELISA or immunofluorescence. In infections, avidity testing can differentiate recent from past infections, as avidity increases over time during affinity maturation. In sum, affinity is like the strength of one handshake. Avidity is the strength of holding hands with both arms. Multiple contacts make the bond stronger. Appreciating this distinction helps in understanding both immune responses and diagnostic assay design. Cross-reactivity is a double-edged sword in immunology. It refers to the ability of an antibody to react not only with its specific antigen but also with structurally similar antigens from different sources. While this can sometimes enhance immune coverage, it often poses diagnostic and clinical challenges. On a molecular level, cross-reactivity occurs when different antigens share similar or identical epitopes, or when the antibody's binding site, paratope, is flexible enough to accommodate similar shapes. This is especially common with polyclonal antibodies, which target multiple epitopes. Even monoclonal antibodies can show cross-reactivity if their specificity isn't absolute. There are several clinically relevant examples. Rheumatic fever arises due to cross-reactivity between streptococcal M protein and cardiac tissue. Guillain-Barré syndrome can follow Campylobacter jejuni infection due to molecular mimicry involving neuronal gangliosides. Dengue virus serotypes show extensive cross-reactivity, which is linked to antibody-dependent enhancement during secondary infections. In diagnostics, cross-reactivity can lead to false positives. For instance, heterophile antibodies can interfere with tests like Epstein-Barr virus detection or pregnancy assays. Similarly, early COVID-19 tests had false positives due to cross-reactivity with other human coronaviruses. On the flip side, cross-reactivity has been harnessed beneficially. Broadly neutralizing antibodies in HIV research can bind multiple viral strains, thanks to cross-reactive epitopes. Cross-reactivity also underlies some heterologous immunity, where past exposure to one pathogen offers protection against another. For students, it's crucial to remember, cross-reactivity is not necessarily a flaw, it's a natural consequence of how antibodies recognize shapes. But its implications in autoimmunity, vaccine design, and diagnostics must be carefully considered and managed. Antigen-antibody reactions form the basis for a vast array of diagnostic tests in medicine. These applications harness the high specificity of antibody-antigen binding to detect either the presence of pathogens, antigens, or the host's immune response, antibodies, in a sample, most often blood, urine, or saliva. Broadly, immunodiagnostics fall into two categories: antigen detection tests, which are useful for identifying acute infections, like rapid strep tests or COVID-19 antigen tests. Antibody detection tests, which are helpful for identifying past infections, immunity, or autoimmunity, such as ELISA for HIV antibodies or ANA tests for lupus. Let's consider some examples. ELISA is used to detect antibodies to HIV, hepatitis viruses, and dengue. It's also used for measuring cytokines and hormones like insulin. Western blot is a confirmatory test for HIV and Lyme disease. Rapid antigen tests using lateral flow technology provide immediate results for diseases like influenza, malaria, and COVID-19. Agglutination assays help in blood grouping and detecting rheumatoid factor. Immunofluorescence is key in autoimmune diagnostics, especially in lupus and vasculitis. Importantly, immunodiagnostics are not limited to infectious diseases. They are essential in endocrinology, e.g., TSH, cortisol levels; oncology, e.g., tumor markers like PSA or CA-125; transplant medicine, e.g., HLA typing and cross-matching; and toxicology, e.g., drug screening via immunoassays. However, interpretation must be contextual. False positives and negatives can occur due to sample handling, cross-reactivity, or test sensitivity limits. Understanding the biological window when antibodies or antigens are detectable is vital. The ultimate message: immunological assays are powerful tools, but clinicians and scientists must understand their principles, limitations, and the biology behind test timing to use them effectively. Now, let's discuss a vital immunological function: antigen antibody complex clearance. Once antibodies bind to antigens, the resulting immune complexes must be effectively removed to prevent tissue damage and to allow resolution of the immune response. The clearance mechanism depends on several factors, most importantly, the size of the immune complex. Large, insoluble complexes are generally cleared quickly by the mononuclear phagocyte system, particularly by macrophages in the liver and spleen. These complexes are opsonized, or tagged, by the Fc region of the antibody and often by complement components like C3b, which enhance phagocytosis. Smaller or intermediate-sized immune complexes can be more problematic. They may remain soluble and circulate in the blood longer, increasing the chance of deposition in tissues such as the kidneys, joints, or vasculature. This is especially relevant in diseases like systemic lupus erythematosus, or SLE, where poor clearance or persistent formation of immune complexes leads to inflammation and tissue damage. Erythrocytes also play a surprisingly helpful role. In humans, they express complement receptor 1, or CR1, which binds to C3b, tagged immune complexes. These erythrocytes transport complexes to the liver or spleen for safe removal by resident macrophages, a process termed immune adherence clearance. Efficient clearance is crucial to avoid immune complex diseases, which we'll talk more about shortly. When clearance fails or is overwhelmed by excessive antigen antibody production, immune complexes can deposit in tissues, activate complement locally, and trigger inflammation. So, to summarize, antigen antibody reactions not only serve to neutralize or opsonize pathogens, but also must be cleared efficiently. This phase is just as critical in preventing collateral damage and in ensuring immune homeostasis. Let's tie the theory back to the clinic by looking at how antigen-antibody reactions contribute to disease processes. These mechanisms can be protective, but when dysregulated, they are often at the root of immunopathology. First, consider type 2 hypersensitivity, which involves antibodies, often IgG or IgM, binding directly to cell surface antigens. This binding activates complement and leads to cell lysis or phagocytosis. Clinical examples include autoimmune hemolytic anemia, where antibodies target red blood cells, and Goodpasture syndrome, where anti-GBM antibodies bind basement membranes in the lungs and kidneys. Then there's type 3 hypersensitivity, where immune complexes form in circulation and deposit in tissues. These complexes trigger complement activation and attract neutrophils, resulting in inflammation. Classic diseases include SLE, serum sickness, and post-streptococcal glomerulonephritis. In type 1 hypersensitivity, although not typically thought of in the same way, antigen-antibody binding between an allergen and preformed IgA is central. This binding cross-links Fc receptors on mast cells and basophils, leading to degranulation and the release of histamine and other mediators. This is the basis for anaphylaxis, allergic rhinitis, and asthma. We also see antigen-antibody interactions in diagnostics. For example, anti-dsDNA and ANA in lupus, anti-CCP in rheumatoid arthritis, and anti-TSH receptor antibodies in Graves' disease. Finally, therapeutic antibodies, like monoclonal antibodies used in cancer and autoimmune diseases, rely entirely on engineered antigen-antibody interactions. Rituximab, for instance, targets CD20 on B cells. So the clinical relevance is immense. Understanding antigen-antibody dynamics allows us to predict disease mechanisms, interpret lab results, and even design treatments. Let's examine this real-world scenario. A 21-year-old female presents with hematuria, blood in the urine, and a positive ANA test. Your first thought might be, what do these findings have in common immunologically? The ANA, or antinuclear antibody, is a hallmark of autoimmune disease, particularly systemic lupus erythematosus (SLE). In SLE, the immune system loses tolerance to nuclear components like DNA, histones, and ribonucleoproteins. Autoantibodies form against these antigens, and the resulting immune complexes deposit in tissues, especially in the kidneys, leading to lupus nephritis. This is a textbook example of a type 3 hypersensitivity reaction, or an immune complex-mediated disease. The antigen antibody complexes form in circulation and then deposit in the glomerular basement membrane, activating complement and inducing inflammation. This leads to hematuria, proteinuria, and potential long-term kidney damage. In this patient, the positive ANA tells us the immune system is reacting to nuclear material. If we were to dig deeper, we'd likely find anti-dsDNA antibodies, which are highly specific for SLE and correlate with disease activity, particularly renal involvement. In a diagnostic workup, other immunologic tests like complement levels, C3 and C4, anti-SM, and anti-dsDNA are essential. Renal biopsy would show immune complex deposition, confirmed by immunofluorescence. This case nicely illustrates how antigen-antibody reactions, when dysregulated, can transition from being protective to pathogenic. It also highlights the power of immunological diagnostics in revealing the underlying mechanism and guiding therapy, in this case, likely immunosuppression to reduce complex formation and inflammation. So remember, this isn't just a theoretical pathway. These reactions are happening in real people with real consequences. Let's now consolidate everything we've discussed with a summary table that organizes key antigen-antibody reactions and their applications. This is especially helpful to help you quickly recall the core types of reactions, their principles, and real-world uses. First, precipitation. This involves soluble antigens and antibodies forming an insoluble complex. The classic example is the ring test or Auchterlonie double diffusion. Precipitation is mostly qualitative and used in research or older diagnostic techniques. Next, agglutination, which occurs when antibodies bind to particulate antigens, such as cells or latex beads, causing visible clumping. This is used in blood typing, the Weidel test for typhoid, and latex agglutination tests. Complement fixation follows. Here, the presence of antigen-antibody complexes is detected by whether complement is fixed and not available to lyse indicator cells. While rarely used now, it was once standard for diagnosing infections like syphilis. Immunoassays such as ELISA, RIA, and CLI are quantitative, sensitive, and widely used to detect either antigens or antibodies in samples. Think of HIV ELISA or TSH measurement. Immunochromatographic tests, like pregnancy tests or COVID-19 antigen kits, offer rapid, point-of-care results. They're based on lateral flow principles using labeled antibodies. Then we have immunofluorescence and Western blotting, which adds spatial or molecular resolution to antigen detection. Immunofluorescence is crucial in autoimmune diseases, while Western blot confirms proteins or antibodies after electrophoresis. Finally, immune complex formation and its clearance, central to understanding type 3 hypersensitivity, have both pathological, SLE, serum sickness, and physiological relevance. This table is a compact way to study before exams and also helps you choose appropriate diagnostic tests in a clinical setting. Don't just memorize, try to understand the principles behind each. Before we conclude, here are the key takeaways from today's session that I'd like you to remember, not just for your exams, but for your clinical and research careers. First, antigen-antibody reactions are highly specific. This specificity is central to both the protective function of the immune system and the diagnostic power of immunoassays. It's why we're able to detect diseases with such precision today. Second, the type of antigen-antibody reaction, whether it's agglutination, precipitation, complement fixation, or neutralization, depends on the physical state of the antigen, soluble or particulate, the class of antibody, and the assay design. Third, affinity and avidity are not interchangeable. Affinity is the strength of a single binding site, while avidity is the cumulative strength from multivalent binding. Both are important in determining the strength and stability of immune interactions. Fourth, Antigen antibody complexes must be effectively cleared. Failure in clearance mechanisms contributes to immune complex diseases such as SLE or post-infectious glomerulonephritis. Fifth, cross-reactivity can lead to false positives in diagnostics or even to autoimmunity. However, it also explains phenomena like heterologous immunity and broadly neutralizing antibodies. Sixth, diagnostic applications of antigen antibody reactions are incredibly diverse, from ELISA and RIA to rapid tests and immunofluorescence. Knowing the principles allows you to interpret results wisely and avoid misdiagnosis. And finally, these reactions are not just test tube phenomena. They occur in the body, influence disease progression, and guide therapeutic interventions. Keep these principles in mind as we transition from foundational immunology into clinical and applied immunology. We've covered a lot of ground today, and I want to leave you with both an opportunity to reflect and a path forward for deeper exploration. First, consider these questions to test your understanding. Why is avidity sometimes more important than affinity in diagnostic testing? How does immune complex deposition lead to tissue damage in lupus nephritis? Can you think of examples where antigen antibody binding is used therapeutically? How would you differentiate between a false positive and a true positive result in a serological test? These are not just exam questions, they're the kinds of reflections that deepen your understanding and prepare you for clinical reasoning. Now for further reading. Janeway's Immunobiology, for a deep dive into molecular immunology and antigen antibody interactions. Abbas and Lichtman's Basic Immunology, a more concise yet clinically oriented overview. Clinical Immunology, Principles and Practice, great for correlating with real-life case studies. WHO and CDC laboratory manuals for understanding practical diagnostic assays, particularly ELISA and rapid testing. I also recommend reviewing research papers on monoclonal antibody therapy, especially in cancer and autoimmune diseases, to see how this basic principle is transforming modern medicine. Lastly, don't hesitate to ask questions. Immunology is complex but deeply rewarding. Mastering the antigen-antibody reaction opens doors to understanding not just tests and therapies, but the very language of the immune system itself. Thank you for your attention.